Good morning, grade 10, how are you? Today, inshallah, we'll complete with chapter 13, lesson 4, part 2. Let's start with our lesson, which is talking about the gene expression and regulation. In the previous lecture, we talked about the gene expression in prokaryotes. Today, inshallah, we will complete with the gene expression in eukaryotic cells. So, how are genes regulated in eukaryotic cells? By binding DNA sequence in the regulatory region of eukaryotic genes, transcription factors control the expression of those genes. So we will talk today about the transcription factors and how these transcription factors will bind to the DNA sequence and regulate the expressions of some genes. We will talk about the DNA genes. The DNA genes, or the genes in the eukaryotic cells, are huge, very large, more complex than the prokaryotic cells. Let's take an example of these genes like TATA box, T-A-T-A box. A T-A-T-A box is a region where we can find the T-A-T-A-T-A sequence or T-A-T-A-A-A that is usually found just before the gene. The TATA box binds a protein that helps position RNA polymerase by making a point just before the beginning of the gene. So we have a TATA box before the beginning of the gene, which will attach, or the transcription factors will attach to it, and then the RNA polymerase will start the transcription process. But before the RNA polymerase to start this process, we need to know that we have another transcription factors that will attach to the enhancer. So we have three different locations on the gene. The first one before the gene, enhancer, and TATA box genes. Okay? Without the TATA box and enhancer, there is no RNA polymerase will attach to the gene. So transcription factors form a binding site for RNA polymerase. Then transcription factor will attach to the enhancer. Other transcription factor will attach to the TATA box or TATA box. Then the DNA will bend to form a binding site for the RNA polymerase to attach to the exact position to start the direction of transcription. So again, as you can see, this is a pent for the DNA with the enhancer and TATA box location and with the transcription factor. So again, gene expression in eukaryotic cells can be regulated at a number of levels. We have multiple levels. DNA binding proteins known as transcription factor regulate gene expression at the transcription level, and by binding DNA sequence in the regulatory regions of eukaryotic genes, transcription factors control the expression of those genes. Some transcription factors enhance transcription by opening up tightly packed chromatin. Other will help attract the RNA polymerase. Okay? Other maybe block access to certain gene. In most cases, multiple transcription factor must bind before RNA polymerase is able to attach to the promoter region and start the transcription, as we mentioned before. All of these points is mentioned. Okay? We have something else called RNA interferences. Okay? For years, biologists wondered why cells that contain lots of small RNA molecules, only a few dozen bases long, and don't belong to any of the major groups of RNA, mRNA, tRNA, or RNA. So we have a small sequence of RNA molecules which is not related to the rRNA or tRNA or mRNA. Okay? These small RNA molecules play a wonderful or powerful role in regulating gene expression by interfering with mRNA. After they are produced by transcription, the small interfering RNA molecules fold into double-strand hairpin loop. So we have something called hairpin loop, double strands made up of small interfering RNA. Then there is another enzyme called the dicer. This is the enzyme, the dicer or the cutter, an enzyme that will cut or dice these double-strand groups into microRNA, miRNA, about 20 base pairs in length. The two strands of the loop then separate, as you can see here. Okay? So this is RNA, okay, interference. It's make up the hairpin loop. The dicer will cut and dice it into small fragments. The fragment is about 20 base pair. Then there will detachment of the two strands or separation of these two strands, and then one of them will bind to a silencing complex which will attach to the mRNA, which complementary base pair sequence to miRNA. One of the microRNA pieces attached to the cluster of proteins to form what is known silencing complex. So we have something called silencing complex. The silencing complex binds to and destroy any any mRNA containing a sequence that is complementary to the microRNA. So again, as we mentioned before, one of them will attach to the silencing complex, and this silencing complex attached to the mRNA, it will stop the mRNA from translation, and that will block the function of the mRNA that fit it, okay? The microRNA sticks to certain mRNA molecules and stops them from passing on their protein making instructions. Once the silencing complex with the miRNA or microRNA attached to the mRNA, which is complementary to it, then it will stop the translation process. There is no protein will produce. So blocking gene expression by means of microRNA silencing complex is known as RNA interference. RNA interference will produce no proteins after it's attached to the mRNA that will fit it, okay? At first, RNA interference seems to be rare events found only in a few plants and other species. It is now clear that RNA interference is found throughout the living world, and that it even plays a role in human growth and development. Okay? Let's move on to the promise of RNAi technology, RNA interference technology. It's made it possible for researchers to switch genes on and off at will simply by inserting double-strand RNA into cells. Then the dicer enzyme will complete the process by cutting the microRNA, which activates the silencing complex. Once the silencing complex is attached to the mRNA, that will stop the building of or the translation of the translation of mRNA or the messenger RNA, and that will not produce protein. This technology help the studying of the gene expression in the laboratory, and it also holds the promise of allowing medical scientists to turn off the expression of genes from viruses and cancer cells, and it may provide new ways to treat, perhaps even cure diseases that it's caused by the genes. And that's all about the eukaryotic gene expression. Thank you for listening today, and have a nice day.